Because our highly productive bioenergy grasses grow very tall, that means that we needed growth chambers that were also very tall. And it turns out that on our campus, there were no existing facilities that could accommodate this. We've custom built stable isotope labeling chambers that house individual plants. And when we're ready to add the stable isotope label to determine what's happening in the plant and in the interactions between the plants and the microbes, we put the cover over the top of the chamber. So what's unique about our chambers is that we have this modular design that we can stack the levels up as our plants are growing. When we designed these growth chambers, we used our existing knowledge about the light spectrum, the light intensity, and the different growth conditions that are needed for our plants to be thriving in this controlled environment, similarly to how they would thrive out in the field. We're also able to provide precisely the amount of water that the plants need to grow productively. And we can add fertilizer in the irrigation lines to provide the nutrition that the plants need as well. Every element on Earth occurs in different stable isotope forms, which means that they vary only in the number of neutrons. This means that they have different atomic mass, but the same chemical properties. We can take advantage of the fact that there's usually a stable isotope form that occurs in very low abundance. So we can experimentally add, for example, for carbon, C13, in large amounts, and therefore we can trace that C13 from carbon dioxide into the plant, into different compounds within the plant, and into the soil, and actually into the DNA of the microbes that are utilizing that carbon from the plant. In this initial round of experiments, we're simply growing the plants and learning how to harvest them and preserve the plant tissues so that we can do the analysis of the specific compounds that contain the isotope tracer. As we harvest our experiments, what we're looking at is specific compounds within different plant tissues so that we can understand where the high-value products such as lipids are being produced and how we may better achieve our targets of increasing the lipid content of our crops. We're also looking for how the composition of the compounds being released from the roots and the composition of plant residues are changing and how that links to the activity and growth of the microbial community that's cycling nutrients in the soil and also contributing to soil carbon storage. Interactions between plants and microbes can be really important. A teaspoon of soil can contain over 10,000 different microbial species, and most of the microbes in the soil at any given moment are not active. This means that it's really difficult for us to determine which microbes are contributing to the important plant-microbe interactions. I'm really excited about this facility because using stable isotope tracers allows us to look specifically at these plant-microbe interactions. And that will allow us to answer questions that we haven't been able to address before.